My name is Dr Emma Burrows and I'm a team leader here at the Florey. Before we begin, I would like to take a moment to reflect on the meaning of place and in doing so, acknowledge the traditional owners on country where we all are today. The Florey Institute is on the land of the Wurundjeri people of the Kulin Nation, and I would like to pay respects to elders past, present and emerging. I would also like to extend these respects to elders past, present and emerging from all the lands from where you are joining us today. We celebrate the traditional owners as the first scientists and deeply respect their sophisticated knowledge systems. During this pandemic, we have found ourselves on country daily, and the value of this connection to our personal health and to those around us has never been clearer. So thank you for joining us today. October is Mental Health Month, and World Mental Health Day tomorrow gives us the opportunity to shine a light on the mental health research here at the Florey. The mental health toll of the pandemic, the restrictions and border closures have been widely recognised, and significant investment in key services to help people right now have been made just this week in the budget. But what about the long game? We need to know what underlies our resilience or vulnerability in the face of stress to know how best to support mental health. The Florey houses a dynamic group of scientists who are committed to understanding the underlying drivers of mental health problems. Before we open our virtual stage to four of our brightest scientists, I would like to introduce Professor Andrew Lawrence, a world leader in addiction neuroscience, who will illuminate why discovery research into mental health is so important. Over to you, Andy.
Thanks, Emma. Good afternoon, everyone, and welcome to our online public lecture series. For those attending who are not familiar with our work, the Florey is the largest brain research group in the Southern Hemisphere. We're in the top 10 of the world's best brain research centres. Our work is critical as diseases of the brain and mind affect us all in some way. In fact, every year, 4.7 million Australians are diagnosed with one of the conditions that we study. As you all know, this pandemic is a chronic stressor with no obvious end. We have had to say goodbye to many rituals, such as handshakes, that are a source of reward and belonging. We're burdened with the stress of financial insecurity and increased caring responsibilities. While we've all been hit by the same storm, we're not all in the same boat. What determines our ability to cope in the moment and in our recovery after this pandemic has subsided? With 1 in 4 people experiencing mental health issues, how will these stresses exacerbate already debilitating symptoms? Are we facing another mental health pandemic? As we wait for a COVID-19 vaccine, the value of medical research is inarguable. We must also invest in a deeper understanding of mental health and in developing treatments. Please join 4 of the Florey Institute's mental health research leaders as they discuss what makes our brain resilient or vulnerable. How do isolation and stress impact us, and how will this affect us for generations to come? The Florey brings world-class researchers together to work towards a common goal of understanding mental health problems and finding better and more accessible treatments. We have diverse groups that tackle this goal across the lifespan, from childhood through to the aging brain and beyond to transgenerational effects. So we have some key questions in front of us. Why is mental health important? Why is it critical to invest in mental health research right now? What don't we know? Well, fortunately, there is now much more awareness of mental health issues in the broader community. My own research interest is in drug and alcohol abuse. You might be surprised to learn that globally, the biggest killer of people aged between 15 and 49 is alcohol. Incredible, isn't it? Remember also that a number of cancers have a strong lifestyle component, maybe linked to mental health, such as smoking, drinking, poor dietary habits with eating disorders, etc. Now, this is just one example of one area of mental health. Today, you will hear about a number of mental health-related research programs that all share a common theme. They are fundamental, basic research into the neural mechanisms that underpin mental health conditions. We contend that such information is vital to enable the discovery and validation of new, more targeted therapies that will be more successful with fewer side effects. Clearly, for a translational pipeline to thrive, we need new basic discoveries to feed into this. At the Florey, we pride ourselves in excellent basic research that has strong translational relevance. Right now, the funding of such basic research in Australia is under threat. Now, I'd like to extend a warm welcome and thank you to our supporters and donors who are with us today. Thank you for joining, and thank you for your generous commitment to our research. As a medical research institute, our work relies heavily on the support of the community and generous donors who recognise the importance of neuroscience as a final frontier in medical research. You too can support our research by spreading the word about the Florey and the learnings that you take away from this series of talks today. If you're not already a Florey donor, we invite you to consider becoming one. All contributions, big or small, are tax deductible and take us one step closer in our quest to find the cures for diseases that affect brain and mind. Another way you can support our work is by leaving a gift in your will. If this is something you would like to consider, please email the fundraising team after the lecture and our bequest manager would be in touch with you. We'd also love to keep you up to date with all the research happening at the Florey and invite you to join our mailing list if you're not already a subscriber. Now it's my pleasure to hand you back to Dr. Emma Burrows and Dr. Christina Perry, who will chair the session. Thank you.
So our first speaker, Katie Drummond, is a final year PhD candidate researching the neurobiological and behavioural consequences of early life stress and also how these relate to anxiety. Today she will shed light on how isolation can change us. Over to you, Katie.
Thank you for the opportunity to present today. I've been asked to discuss the much talked about question, what does isolation do to us? And because of the COVID-19 outbreak, almost every one of us is more isolated than ever before. This will likely have implications on our mental health now and in the future. So we need to know what isolation does to us. If not for us, we need to answer this for others. Remember, 1 in 5 Victorians experience a mental illness or disorder in any 1 year. We need to understand the impact of isolation on their mental health conditions. It is also important to understand the impact of more vulnerable parts of our community. For instance, 3 in 4 of all mental health problems appear in people under the age of 25. We have been in lockdown for more than 5 months now, and these restrictions in some form will persist for some time, making it even more important to understand the ongoing impact on people even without a mental health condition, particularly with so many of us facing other pandemic-associated stress. There is a lot of discussion about social isolation in the media, so I want to define it to make sure we're all on the same page. It is an objective reduction in social contact, and it's not necessarily the complete elimination of it. If we look at this image and imagine that one dot is a person like you or I, and that each line is a physical interaction, and we implement the COVID-19 containment measures currently in place, then you can see that these measures reduce our connections with each other. By reducing it, it likely leaves us, by definition, to be more isolated than before. Social isolation has a wide array of effects, and its impact really does vary from person to person. Arguably, the most discussed is an increase in loneliness. Another is a reduction in social support, and also a reduction in physical activity. All of these and more start to jeopardize our mental and brain health, and data from previous pandemics support this. When we jeopardize our brain health, we jeopardize our health overall, and social isolation is associated with an increase in mortality. Paradoxically, if we are exposed to an infectious disease, evidence suggests that social isolation might make us more likely to get infected and more likely to have more severe symptoms. To understand what isolation does to us and why, we need to understand the effects of isolation on the brain itself. We can model these effects quite well preclinically. We aren't the only social animal. In fact, many animals rely heavily on interpersonal connections for mutual health benefits. Since the 1960s, scientists have been characterizing the effects of social isolation on brain and behavior. Here is a graph demonstrating this. And you can see we're now in 2020, and we can expect around 8,000 preclinical studies on this topic. The most common way of modeling the effects of isolation preclinically is with rodents. They too have social networks and interact with each other and rely on these interactions to have a healthy brain, just like us. The basis of a preclinical isolation model is simple. We enforce social distancing. Just like us right now, they can smell, hear, and see their peers, but they just can't physically touch. Our lab is interested in using this preclinical model to understand what isolation does to our brain in terms of anxiety. To understand its impact, we answer this question in 3 separate steps. First, we investigate the effects of isolation on a wide range of behavioural tests. This allows us to know if social isolation increases anxiety and who might be at risk. Second, we investigate how treatable or reversible these effects are. This allows us to gauge the actual impact of social isolation on the brain and anxiety-like behaviour. Finally, we aim to use our research to improve our understanding on the impact of isolation on the brain. We can test solutions that can improve outcomes for our brain health and therefore our mental health. I want to focus on one impact that social isolation has on the brain, and that's its impact on anxiety-like behaviours. Since the 1960s, researchers like us have worked to understand if isolation changes anxiety-like behaviour. Using an array of tests, they have examined the impact of isolation in adulthood and in adolescence. First, I'll show you the effects of isolation on anxiety in adulthood. In this image, I've included 10 separate measures of anxiety-like behaviour. And you can see that some studies find increases in anxiety in red, but others do not, suggesting that the healthy adult brain may at least somewhat be resilient to the effects of isolation on anxiety, but we don't really understand what makes some resilient and some not. If isolation occurs early in life, when the brain is still developing, these effects are more profound. High anxiety is indicated again in red. What's worse is that some of these studies report that after reintroducing physical contact with peers, anxiety remains persistently high, meaning adolescents are vulnerable to the immediate and long-term effects of isolation on anxiety. With this knowledge, we can move on to the next step, to understand how reversible or treatable the impact of isolation is on anxiety. One way to do this is by examining how easy it is to regulate fear or learn not to be afraid. If this can be done well, the impact might not be so great. We can examine the impact of isolation in adulthood and in adolescence. We can train animals to regulate their fear, somewhat like an animal therapist, and then a few days later we can test to see how effective it was. Are they scared or are they not? We find that isolation in adulthood doesn't affect this outcome, and similar levels of fear are displayed. However, it's a different story if isolation occurs early in life. Early in life, isolation results in high levels of fear, meaning it's more difficult for them to regulate fear. To understand this, we need to its impact, we need to further see the long-term effects of this. After being isolated in adolescence, we can reintroduce physical contact and then assess the effects later in life in adulthood. And what we find is that early life isolation has long-term effects on fear regulation. Fear is still high in adults that have been isolated in adolescence, meaning adolescents are vulnerable again to the immediate and long-term effects of isolation. Preclinical models predict that isolation early in life both increases anxiety and may make it harder to treat. We can then move on to the final step, to test what will improve outcomes. I mentioned before that social isolation often results in us exercising less. So we wanted to see if increasing physical activity early in life can protect our brain from the effects of social isolation. We replicated our finding that isolation impaired fear regulation in adolescence. But when physical activity was increased, which I'll show you now in white, the brain was protected. and isolation no longer affected the ability to regulate fear. This provides some clear evidence that physical activity early in life is protective for the brain and could improve mental health outcomes. Of course, we need to understand the biological mechanisms underpinning this, and that's a challenging task. So the adolescent brain is growing, and circuits that regulate fear and anxiety are taking in information about our environment to mature. And one of the ways it does this is that these cells in a region in the brain called the hippocampus. And we found evidence to suggest that these cells mediate the protective effects of exercise in the developing brain. How they are doing this, we don't know, but we'd love to find out. As I'm out of time, I want to take us back quickly to the question put to me in the first place. What does isolation do to us? This is important for every one of us to understand, but my take-home message for you today is that the mental health of vulnerable populations are more likely to be at risk: the young, the old, and the mentally ill. Preclinical research can help us combat this, modelling future outcomes and identifying evidence-based solutions. Thank you again for the opportunity to speak, and I am just one of a team of researchers who are trying to understand how environment affects the developing brain. Thank you.
So our second speaker, Dr Jess Nithianantharajah, heads the Synapse Biology and Cognition Laboratory. Jess's work looks at how genes can shape complex mental processes and therefore our mental health. Over to you, Jess.
Thank you so much, Emma. It's a really great pleasure to be here, and thank you all virtually out there for really joining us. So what I want to share with you today is how understanding our own biology through understanding our genes is actually starting to shed some insights into how the brain shapes our behaviour and therefore our mental health. So the genomic era of being able to decipher the genes that make us who we are has really opened the floodgates for how we can apply genetics in medicine. So across a wide variety of medical conditions, genomics has really transformed the way that we now diagnose and treat rare and common diseases. So the human genome is the complete assembly of DNA, around 3 billion base pairs, that makes each one of us unique. And while DNA holds the instructions for building the proteins that carry out these functions in our cells, we know that genes don't work in isolation and that gene expression can be modified by the environment. And this is the process of epigenetics. So the epigenome is made up of these chemical compounds that attach to the DNA to actually impact how those instructions are carried out. And so a key message that I really want to convey today is that our biology and our mental health are both on a continuum. Now, it's important to recognise that we all harbour genetic changes or mutations in our genetic code that really make us unique and is actually the underlying factor that drives evolution. So this is just to say that I think when we think about the concept of genetic mutations, it is often has a negative load associated with that. But a genetic mutation is just simply a way of characterising that there are changes in the genome. And it's just these natural changes that actually are the underlying basis for creating diversity and adaptability. So in thinking about our own genetic vulnerability to disease, we all lie somewhere along a spectrum, let's say, or a continuum. For some of us, the functional consequence of mutations or changes in our genes or genetic load is really low, while for others, that functional consequence of changes in our genome or the genetic load is actually quite high. Now, similarly, our mental health is also on a continuum. So on the one side, for example, you have healthy functioning where you can navigate your way through life with relative ease. And on the other, for example, you have where daily functioning is actually severely impacted, and obviously a range in between. And so how our biology interacts with the environment can actually shape and shift where we find ourselves along this scale. So for example, if someone with a really low genetic load, for example, experiences a stressful event, and you'll hear about these today, what happens is it's likely going to shift that person to have some distress and have impacts on how they cope and function. In comparison, if you have somebody who has a genetic load that's moderate, for example, that same environmental stressor is going to shift them further down for it to have a more severe impact. And clearly, if you have a higher genetic load, you may face daily changes as it is. And so genetic studies over the last 10 to 15 years have really started to unravel the complexity of the genetic basis of a range of mental disorders, really providing insights that, and sort of shown in this schematic here, that as gene vulnerability and mutation load increases in individuals, so does the likelihood that that individual will show increased symptoms. So there's a host of different types of mutations or gene changes that we can all have. You can have, for example, single mutations that are quite severe to less severe mutations, and you can actually harbour multiple mutations. And what this does is it gives us all a genomic risk, and that genomic risk is then subsequently influenced by developmental variation during brain development, and this can then lead to changes in brain function, resulting in a wide variety of disorders. And so over the years, despite a wide number of mutations that have been uncovered across mental disorders, one of the interesting molecular pathways that gene mutations seem to converge on actually involves connections in the brain. And this is where we've been really interested in understanding. So the connections in our brain, or synapses, are really critical for how the brain communicates and ultimately relays chemical signals to each other, and therefore really underlies how behaviour is controlled and regulated. These connections form discrete circuits in the brain and allow us to control our actions and ultimately switch between appropriate behaviours when changes in the environment happen. So to understand the importance of genes at these connections, we can use mouse models with these gene mutations, modelling exactly those that are found in individuals, as a powerful tool to really understand some of these shared biological pathways that are disrupted. And while at the clinical diagnostic level, when you look across a host of different mental disorders, they appear to be quite different. And they do, of course, have symptoms that are quite unique to them. But the reality is it actually looks a little bit more like this, whereby there's actually a lot of overlap between them, with behavioural and cognitive deficits that are really at the heart of all these disorders. Now, it's important, I think, to appreciate that all these disorders are, again, on a spectrum, in that there's a wide range and severity of symptoms that people experience. And in addition to that, there's actually high comorbidity with these disorders. What I mean by that is that individuals most often have a diagnosis of multiple disorders. And what's additionally interesting, actually, is that even two people that are diagnosed with the same disorder are never exactly identical and have their unique strengths and challenges. What this does, it really highlights the complexity in understanding these conditions and the need to think about addressing these at an individual level. So towards understanding how genetic mutations can impact behaviours that are relevant to mental disorders, our lab has been really working with these advanced behavioural tools in rodents using these technology for over 12 years now. And these tools, where we can train rats and mice to be able to use touchscreens, has really become a valuable tool for how we enable translation. And the Florey is one of the largest national sites really employing these sophisticated tools. What this really allows us to do is compare cognitive testing in both humans and mouse models. So what we've been able to previously show is not only can we utilise similar tests in humans and mice with the same genetic mutations to understand how that mutation impacts behaviour across species, we've also been able to utilise the exact same tests in our mouse models and in individuals. And just recently, we've shown that we can use these tests to really dissect and measure changes in motivation in one of our animal models, providing these really important molecular insights into how these processes are regulated in the brain, and therefore providing opportunities to address the biology of conditions such as depression and mood disorders. So just globally, one of our overarching goals is to really identify how disruptions in distinct genetic codes that govern connections in the brain lead to specific changes in cognitive behaviour. And what this will ultimately enable is pathways towards personalised medicine. I think this is a theme that you'll hear a lot about today. And ultimately, this is really what's needed to advance how we tackle mental health disorders. So that going forward, what we can do is identify how genetic vulnerability leads to disruptions in select behaviours that, as I've alluded to, are shared across disorders, and therefore develop targeted treatments and options that allow us to treat the cluster of symptoms that an individual presents with, rather than thinking about treating everyone with a broad disorder in the same way. And so lastly, knowing our genetics might make us vulnerable, but how can we protect our mental health? And so this idea of the mental illness jar is from a blog by Janine Austin. And I think it really nicely illustrates that if we looked at ourselves and our mental capacity as jars, for example, that both genetic and environmental factors play a role in filling that jar. So in order to be actively experiencing an episode of illness, the jar must be filled to the top. And different people's jars can be filled with different amounts of genetic and environmental vulnerability factors. So for some of us, that genetic load is low, so you have more room for environmental impacts, for example, while for others, that genetic load is high. So while we can't make the jar itself any bigger, what we can do is create more room by stacking lids or protective factors. And some of these include sleep, nutrition, exercise, good social support, and also importantly, medication, so that that jar fills up more slowly. I think it's important to reflect that on our own individual capacities and the needs, so that we identify how we can adaptively work with triggers in our environment to really help maintain positive mental health. So I want to leave you with that and say, I hope that we continue to advance how we diagnose, manage and treat mental health conditions, and that we continue to push discovery-driven approaches so that one day we view and treat mental health conditions in the exact same way that we now treat physical health conditions. Thank you very much.
Thank you, Jess. So I'll just introduce myself. My name is Christina Perry, and I'm really delighted to be able to introduce our next speaker, my colleague, Dr Erin Campbell, who, like myself, is a behavioural neuroscientist and a team leader in the Addiction Neuroscience Lab here at Florey. So Erin's research is focused on understanding how neuropsychiatric disease states change the brain's chemistry, structure and function. Erin and her team hope to identify novel brain areas and circuits that future clinical studies can target in people with alcohol use disorder who are seeking treatment. So I'll hand you over now to Dr. Erin Campbell to talk about how does stress impact on existing mental health conditions and what interventions we are looking at developing.
Thanks, Christina. Thanks for the introduction. Today I'd like to talk about the interaction or the effect that stress has on our mental health, which I think is quite topical, particularly given the current global pandemic. And I'd really like to focus on the individual, because how each of us as individuals respond to stress or cope with stress ultimately has an impact on our own mental health. So when we talk about mental health disorders or mental health conditions, we're talking about disorders that are chronic, so occur over a lifetime, and they're relapsing in nature. So when I talk about relapse, what I mean is the recurrence of a pathological behaviour after a period of abstinence. And these disorders or these mental health conditions are brain disorders. So I have a few examples here, which a few of the other speakers have already touched on. So anxiety, depression, obsessive compulsive disorder, obesity, and our lab is particularly interested in substance use disorders. We know that relapse, or one of the most important factors preventing the successful treatment of mental health conditions, in particular substance use disorder, is relapse. And this is not a new phenomenon. So we know that up to 90% of individuals with substance use disorder relapse, which is a horrific statistic. And I actually have a recent example just down below here. So Dax Shepard appeared in the media for relapsing to opioid use or prescription medication use after 17 years sober. which is horrible. So one of the main goals of our lab is to prevent relapse. However, before we can focus on the prevention of relapse, we need to understand what the causes of relapse are. And there are several factors that promote relapse or cause relapse. And one of the main factors is stress. So for example, if we think about having problems at work, having relationship issues or relationship breakdowns, and having emotional, psychological, or continuing emotional, psychological issues, all these factors, all these stressful factors can impact our relapse vulnerability. And our lab is particularly interested in understanding the mechanisms, the neural mechanisms of stress and how that's linked with mental ill health. And I'd just like to focus on this schematic. So these are the three, having trouble with my mouse, the three sort of factors that influence how we respond to stress. So we have our biological sex, we have the type of stressor, so whether it's physical or psychological in nature, and we also have the age of the stressful event. So whether it occurs in early life or adolescence, as Katie mentioned, or whether it occurs in later life. And unfortunately, COVID has brought about many new stresses and also enhanced amplified existing stresses. So if we think about financial distress, for example, financial hardship, and these stresses have been both a direct consequence of the virus itself and an indirect consequence. So due to things like isolation, feeling overwhelmed, changes in daily routine. And importantly, people with an existing mental health condition have a really complex relationship with stress, and their response or their ability to adapt to stresses may ultimately increase incidence of mental health conditions and relapse rates. So I would like to give a few examples of how COVID as a stressor can cause relapse. So if we take depression, for example, so changes in our daily routine caused by lockdown and changes in social rhythm can really impact people with depression and cause relapse, in particular, depressive-like behaviours. And if we think about individuals with substance use disorder, being in lockdown, having limited access to perhaps those abused substances or medications can cause severe withdrawal symptoms. So things such as what are we talking about here? Delirium or seizures even. So it's just really important that we think about the consequences of the lockdown and of COVID in general on our mental health, and even in terms of what the lockdown environment promotes. So for example, we've seen increases in alcohol consumption, increases in online gaming and online gambling. But the two main points that I'd like to highlight is that there's individual variability in how stress impacts us, which is caused by the interaction of sex, of the type of stressor, and the age of the stressful event. And these factors ultimately interact and lead to variability in the vulnerability to relapse. And psychiatric disorders are disorders of the brain. So we know that these conditions rewire the circuits in our brain, which ultimately alter brain function. And now I'd just like to talk about some of the recent data our lab has collected. So if you remember, I said that we're particularly interested in predicting relapse, or predicting relapse vulnerability, which is actually no easy task. So ultimately, we'd like to be able to intervene early and predict or prevent a relapse episode before it occurs. So we know that there's huge individual variability in the likelihood or the propensity of an individual to relapse. This is caused by stresses in our environment and our ability to cope or to respond to these particular stresses. And our lab's particularly interested in relapse to alcohol use. And we study this in rats and mice. And we know that in humans, taking a person-centered approach to treatment is very important. And this is what we try to model in our rat and mouse models. And so you can see some, I've got some data just here on the left. So this is in male and female rats, and this is our measure of relapse-like behaviour. And you can see that there's huge variability in relapse behaviour. So we've got animals that are very likely to relapse versus animals that aren't. And we see the same variability in males and females. And actually what's interesting to point out is the sex difference that we see. So females are less likely to relapse, female rats are less likely to relapse. And this is all in response in response to a negative consequence. Actually we do have some new data just hot off the press where, remember I said we were interested in predicting relapse vulnerability? So we have actually found that, at least in female rats, the prior response to a negative consequence is able to predict relapse-like behaviour. We're still trying to find factors that predict relapse in male rats. That's ongoing research. And then just over here on the right, so I remember I said that alcohol use disorder and mental health conditions are conditions of the brain. So here we have some data where we're looking at a particular brain region called the anterior insular cortex. And what we actually showed was increased activity in this particular brain region in high relapsing rats. And what we can do is, if we actually block the activity, so functionally block the activity of this brain region, we can prevent relapse in these high relapsing rats. So we're not finished yet. We still have a lot more work to do. We're really interested in I guess, delving deeper and understanding the role of individual neurons in relapse-like behaviour and the individual circuits that are involved. And ultimately, we'd like to get a better understanding of alcohol use disorder and the addictive process, which will ultimately lead to more effective therapeutic strategies for the prevention and treatment of substance use disorder. And I just have a few acknowledgements just at the end here, but thank you.
Thank you, Erin. So our final speaker is Dr Terence Pang. Terence is a postdoctoral researcher whose work embodies a unique multidisciplinary approach by combining behavioural neuroscience and reproduction biology. He aims to uncover how stress contributes to emerging trends in mental health across generations. Dr. Pang will now examine what are the long-term impacts of this stressful event.
And good afternoon, everyone, and thank you for taking the time to listen and learn about the research that's going on at the Florey. So this pandemic has forced a very different way of life upon all of us, with many restrictions to our physical movements and social interactions. And this has generally led to an increase of stress across the board and contributing to heightened anxiety and even incidences of depression. So many of you would be familiar with this consistent message that maintaining good mental health is vital for keeping healthy in the present time. But while developing an effective vaccine remains the most pressing issue at this moment, some of us have started to think about the long-term effects of this pandemic. Now, you've heard my colleagues talk about how stress affects us individually, but my research focuses on its legacy on mental health across generations. And this legacy refers to the relationship of the health of one generation and the processes that ultimately shift or dictate the health outcomes for their children and future generations. So this is a relatively new concept to the medical sciences, but evidence is certainly increasing that parental or even grandparental factors such as their dietary choices, their level of nutrition, history of smoking and even alcohol consumption, and perhaps even experiences of traumatic stress, all these can significantly affect the physical development and subsequent health of our offspring. So here at the Florey, my research focuses on the transgenerational effects of stress, specifically paternal stress. And while we live under this cloud of unabated worry over our health, our children's education, job or housing security, what I want to figure out is what's going to happen many years down the road, and how could our current experiences potentially influence the health of our children, and perhaps subsequently even their children. So my team has been studying a unique animal model of chronic stress that I had developed here at the Florey. For this model, we increase the level of stress hormone circulating in the mouse in a precise and measured manner over a fixed length of time, usually about a month or so. We can then use these mice for breeding. We obtain the offspring, and then we have the capacity to study the behaviors of these offspring once they reach adulthood. And what we found was that despite the stress we impose not causing the fathers themselves to change their behaviors, so that's consistent with the idea that we are only imposing low levels of stress, their sons, under no additional stress themselves, were in fact more likely to display behaviors reflective of high anxiety. So in these two graphs that you see here, showing data from two common tests for rodent anxiety, if you focus on the white bars, they indicate that the sons of stressed fathers are more apprehensive and spend significantly less time exploring novel and exposed spaces, which rodents naturally tend to do, being curious and inquisitive animals. Interestingly, across all the behavioral tests we conducted, the daughters of stressed fathers did not display any alterations to their anxiety level, indicating that we are dealing with sex-specific effects. Now, we have also found that going on to the next generation, the grandsons were more likely to display higher risk-taking behaviors along with some features of depression. So this is a demonstration that there is a transgenerational effect of paternal stress. Now, the most exciting aspect to our work was that we were able to identify a unique molecular signature in sperm that we harvested from the stressed mice. And this signature appears to convey information about the father having been stressed across the generations. So in this case, we are talking about a small group of molecules called microRNases that are present in sperm. And just note that they are in addition to the heritable DNA component. And our study found that there are 101 microRNAs that are expressed at different levels in the sperm of stressed mice. And we currently have projects ongoing looking at how this potentially changes the early development of the offspring and their brain function in future. But while we continue to more comprehensively profile other molecular properties of sperm that may be affected by chronic stress, my South Australian collaborators are now using our discovery and studying human clinical samples for this exact signature. We think that by refining our ability to take molecular measures in men to gauge their history of stress and their exposure to stress, this could possibly increase self-awareness and inform better reproductive choices in future. So the big question is, how is stress changing the molecular signature of sperm? Well, we don't have a definite answer, but we came up with a theorized model. So it turns out the male reproductive organs have the second highest density of stress hormone receptors outside of the brain. And these are expressed right across the epididymis, which is the organ that sperm mature in after they leave the testis. So the theory I formed was that the epididymis is able to respond to stress and the increasing levels of stress hormones that circulate in the blood. And this signals to the organ to convey that information to the sperm, possibly through secreted nanoparticles that are called epididymisomes. Now, these particles are then absorbed by the sperm to be ultimately transferred to the fertilized egg and the next generation. And we can now visualize this process in work beautifully done by my collaborator at Newcastle University. So here you see the mouse sperm that have been incubated with these epididymisomes that are loaded with green fluorescent particles. And over time, you can see the green signal accumulating in the sperm head. Now, unfortunately, due to limited resources, I've been unable to test this in our animal model, but I'm pleased and encouraged by recent published studies from this year from independent teams of American researchers that have firmly supported this model I proposed. However, it's not all bad news. So in our latest published work, we have found that shorter exposures of stress do not carry this form of intergenerational risk. And what that does is really emphasize the importance of stress management. So what can we do to limit our experiences of stress, and what aspects of our lives can we modify? So we've done separate work into stress mitigation through exercise. So here you see that the sons of exercising fathers that engage in voluntary wheel running were less likely to display aversion and were more likely to explore that novel, open and brightly lit space. So I want to integrate this information to formulate new research questions as we try to break this intergenerational legacy of stress. And we're now beginning to explore how other lifestyle factors, such as the level of social and cognitive stimulation, and even dietary choices weigh into this conversation. So I think it's important to also mention the children who have been affected in their own way during this pandemic, with restrictions to their social and physical activities, both of which we know are crucial for normal behavioral and emotional development. And across the past months, the demand for children's support services have spiked nationwide. And we're really interested to find out if there are any long-term consequences for children who experience this excess of worry, anxiety, or fear. Now, it's clear that trauma and stress can leave an imprint on children. So research into 9/11 have found that traumatic stress can even initiate effects during pregnancy. So in women from the New York region who were in their third trimester when 9/11 occurred, their infants at 9 months old were already found to show greater distress to novelty. And a separate study reported higher rates of clinically significant behavioral problems among preschoolers in the lower Manhattan district. Middle and high school age children who experience emotional distress and trauma up to 3 years post 9/11 have in fact continued to show symptoms across the years. So this really demonstrates the magnitude of the problem that we potentially are dealing with. And the reason for our current concern is that animal modeling has found that poor social development negatively impacts on brain development, increasing the risk for poor physical and behavioral growth. So the intergenerational effects of stress experienced by children or even pre-adults remains largely unknown. There haven't been many studies of that. But what we're trying to do here is to play the long game. And these are the types of research projects and questions that we're fighting to initiate and conduct here at the Florey. What my team has begun doing is adapting our animal models of early life trauma to begin to investigate the possible multi-generational consequences that stem from these social and physical challenges during childhood. And this is work that's ongoing, so I'm not going to present any data today, but the earliest indicators do suggest that there are intergenerational effects. And these are very distinct to what we've observed in the adult chronic stress models. So please watch this space as we continue to work on this project, and hopefully the next time I'll have some good results to report on. So I'd just like to summarize and conclude by saying that, well, this pandemic will leave a legacy on our mental health, but the full extent won't actually be known for several decades. Both adult and children are at risk, but they're likely to be dealing with different problems in themselves. So interventions need to be tailored to the different age groups to meet their specific needs. And finally, I just want to emphasize that mental health is a complex issue. There are different sources and different levels of stress, and you've heard about individual differences in resilience and susceptibility. So we really need to develop a wide range of management approaches to ensure that we take care of as many people who need help as possible. So thank you for listening.
Thanks very much, Terence. I'm sure you'll agree with me that we have some outstanding mental health research here at the Florey. These 4 vignettes have shown you that the effects of social isolation may be particularly profound for adolescents and could have a long-lasting impact. They've also shown how disruptions in the genetic code that governs connections in the brain can make you more vulnerable to mental disorders. How treatments for stress-related disorders need to focus on the individual. There's no silver bullet, no one-drug-fits-all model. And last but not least, you've just heard that at a time when our way of life is greatly challenged, it's more vital than ever that we have a clear understanding of the legacy of this pandemic on our mental health and that of our children and future generations. So now I'm pleased to hand back to Emma and Christina to host a Q&A session. Thank you.
Thank you, Andy. And please join me in giving our researchers a big virtual clap and messaging us through your feedback. So thank you all for sharing your fascinating work with us all. Now we have had a number of questions in the Q&A section. Some of these are on issues that are possibly a little bit sensitive for us to deal with, especially with so many in the audience and the fact that we don't know the current impacts of the pandemic and none of us are clinical doctors. However, we do have some other questions which we would like to open up to our panel, and I will start with some questions for Katie. Katie, someone was just wondering what gender differences appear when modelling isolation in rodents, and also whether adolescent rodents are susceptible, that adolescent rodents were susceptible where adults were not, and what's the equivalent age of that in terms of human teenagers?
Thanks for the question. So you raise a really good point talking about gender differences. In preclinical research, there's a lot of bias towards males and the effects of stress on males. And increasingly, we're looking at sex differences in relation to stress. I did look at the effects of stress in males and females, and we found the effects to be more profound in males. This doesn't mean that females are unaffected. It's likely that other domains of behaviour have been affected in females as well, which is why I didn't want to dwell on that too much. And if we look at the effects of loneliness and the effects and mental health effects on adolescents in previous pandemics, we see a similar sex differences, with males more likely to suffer from anxiety-like behavioural changes, and there's reports that females are more likely to have depressive-like symptoms. So hopefully that helps answer your question. And just to highlight, we haven't really delved deep into understanding gender differences or sex differences. So I can't give a very informative answer on that. In terms of the second question about the equivalent human age, this depends on which researcher you talk to, but approximately 8 years of age to up to 24 years of age or 25 years of age is adolescence. Rats and mice develop their brain very quickly, and so it's quite hard to have an exact pinpoint for you. Hopefully that answers your question, and I'll hand it back to anyone else who wants to comment.
A nice segue, Katie. Actually, 2 questions for Terence. Terence, you focused on the male germline in your research, and there's a question that's come through in particular about what happens through the maternal germline. And you did mention that there have been studies in people after 9/11. Could you illuminate what happens in mice?
Yeah, thanks for the question, Emma. I've actually just replied to one similar question in the Q&A. So we know that when you actually stress a mouse model during pregnancy, the pregnant dams respond appropriately by changing their maternal behaviors that they display towards their pups. And that can be regarded as a positive or negative thing, because you certainly don't want the animals to start over-mothering their infants. And this is quite an interesting parallel to some of the over-mothering behaviors that we see in various different cross-cultures as well. So it's difficult to translate something this sort of behavior directly from mouse to humans, but we certainly see some levels of some parallels. But I think that the stress that we subsequently study in humans is going to be quite complicated, and it's going to be quite different from what we observe from the mouse models, and that requires a whole different avenue of research. Very good question.
Thank you, Terence. I've got a question here for Erin. Erin, how long will the burden of COVID be on mental health last, do you think? And how can we help flatten the curve for mental health disorders?
Yeah, that's a very good question. Look, it could go on for quite a long time. I mean, Terence presented data from 9/11, I guess decades after. But at the moment it's undetermined, but we do know that the impact on mental health is quite dramatic. And in terms of helping to flatten the curve, I guess having good coping mechanisms, understanding how you respond to stress and how you cope best, getting help if you don't know how to cope, and I guess ultimately improving our fundamental understanding of mental health conditions and how they're linked with pandemics or stress, and yeah, will definitely help.
Christina, can I add some comments to that? Yeah, I mean, look, absolutely. I think that's been one of the themes here, isn't it? In that with COVID, we're dealing with the direct impacts of that right now. And as Andy alluded to at the start, the likelihood that COVID will have both a direct and indirect consequences will be something that we'll have to very closely monitor as we sort of go forward. One of the things I've been reflecting on, and so, coming back to the direct, we think about the indirect in terms of obviously the stresses, the changes in lifestyle, to jobs, to security that have been impacted, and we can see, and obviously with some clinical colleagues, we've just shown recently that, COVID is in the brain, can enter the brain as well. So again, it's going to be a bit of a long game, as we keep saying. But something that, coming back to mental health, that I think for us, the way that we've dealt with COVID can maybe help, or, sort of help us think about how we try and look at mental health as we go forward, is to think about identifying individuals with symptoms and having a structure in place that people can go to. I mean, this is true for COVID in terms of, looking at their respiratory and or immunological compromise. And I think that is something that we have in place. But for mental health, I think we could provide that more broadly as well. So one is about the identification, people who identify with having mental health symptoms to come forward and be able to go to places where that is then recognised and there's management put in place. The second aspect of that, exactly as we're doing for COVID, is the scientific research that's driving the development of vaccines. And I see that as being a parallel thing for what we need to do in mental health as well. So not only do we have to have two things, which is support services that help people manage what, in a situation where we don't have all the answers, support services can help manage people with the current symptoms. At the same time, funding that pipeline that enables the scientific discoveries to develop better treatments. And I think those two approaches have been something that are core pillars of how we address medicine more broadly. And those two core pillars, I think, still should be exactly how we approach mental health. Obviously, noting that it's far more complex. COVID, we know the virus. And as we've talked about today, the cause of mental illness is obviously far more broader. But that just suggests that we need to invest more in all those aspects than we would in some other conditions, for example. But I think the strategies that we use currently are essentially potentially the same strategies that we should push for with regards to cracking mental health.
Yes, there's been a lot of talk about the jar analogy, and a lot of people in our chat are saying this resonates with me, and I can say the same. I thought it might be nice for us to end off our session with all speakers perhaps talking about what they think could be one of those environmental factors that could be something we invest time in. I know that the pandemic has been hard for all of us in many different ways, and sometimes you just don't have the capacity to be thinking of multiple things. So if you had to pick one thing that you could leave our audience with, what would you pick? We'll start with you, Jess.
So let me, this is about what is one thing you can do for yourself during this time that helps promote your mental health. I think this is something that there's probably been a lot of conversation around, and this has been very true for me, which is, it's hard. I mean, I've got a young child as well, but it's about carving out time for yourself, which taps into aspects of meditation. I think that one of the things I've realised during this time, and it's true in this era, there's a lot of noise in the world. There is so much noise that sometimes it's almost deafening. But I think that's where you have to dig deep and find that time to have quiet moments for yourself, to be reflective, and to think about, the really core values and needs that are really important to you. And just start with that, just to start with those and make those the priority for your life, and to cut some of that noise. And I think that is key.
What about you, Terence?
I think the one bit of advice I could share is, and I've been thinking about this myself, is I know that staying physically active is going to be important throughout this time. It's great stress relief. But at the same time, it's not just about jumping on that treadmill or, putting your Ks in and just going round and round the park or the track or something. I think let's change it up a little bit and maybe take a walk through a suburb within your 5K limit right now, obviously. But walk in a different environment and take in all the new sights and sounds and smells as you're getting your physical activity. I think that combination could potentially be really powerful for the mind.
Erin, did you have anything to add?
Yeah, I think I like that response, Terence, getting in touch with nature. I don't know if many people do that anymore. But yeah, I think taking the time to learn about yourself and learn what works for you as a coping mechanism. So yeah, whether that is exercise or meditation, mindfulness, etc. So just make sure you take the time out and think about how you cope and how you react to stresses or the pandemic and what will benefit you most.
Thanks, Erin. And Katie, did you want to add anything to that?
I guess I completely agree with Terence. It's really important to go outside and engage in physical activity. And some of the work that we're showing is it's really important for children and adolescents to really go outside and engage in physical activity and not just spend all their time on the computer or in front of an iPad. So yeah, that's what I would say.
Can definitely resonate with that one, Katie. Thank you. Okay, well, we'll end there because I am aware that we are running a bit over time. So thank you once again to our stellar panel and to all of you who've joined us today. We really appreciate your interest in the Florey's work. And we'd also like to extend a special thank you and acknowledgement to all those who are living with mental ill health that have joined us today. So I do hope that we leave you with a sense of how discovery science is the force behind innovation and development in mental health treatment. So here at the Florey, we will continue to use evidence-based research to drive advances in this area. And as we uncover and unravel the biology behind the long-term effects of stress, we also will learn how to manage and treat these effects, as well as what types of factors might confer resilience, which is really important. So thank you so much for joining us in our journey. We do hope that we've given you some hope for what the future might bring. Please don't forget to email or call our fundraising team if you have any questions. We weren't able to get to all of the questions today, unfortunately, but feel free to reach out to us if you do have further questions. And of course, if you'd like to donate to our research, the email is fundraising@florey.edu.au. It's just up on the screen there now. So thank you once again for joining us, and I hope that you and your family are staying safe and healthy, and I look forward to keeping you informed about the Florey's research.